Human beings are one of the most dominant and destructive organisms on the planet. Humans become the apex predator in every ecosystem they enter. But there is one thing not even they can escape or destroy, and that is cancer. Cancer has always been a part of human beings' life history. Cancer in humans is common because of how long we live and how we live. Cell senescence, as described by Medawar, is the decline of bodily facilities and sensibilities and energies. Cells senesce when they proliferate, and when they proliferate, they can mutate, which eventually can lead to tumor genesis. Another thing that can lead to this cascade of mutations is known as carcinogens. The carcinogen we will be discussing today is benzene, and because of how benzene is produced and metabolized by the body, humans are not the only dominant species that is affected by it.
Benzene is an aromatic hydrocarbon that is considered to be a group 1 carcinogen, meaning it is carcinogenic to humans. It is known to be the causative agent of acute myeloid leukemia, as well as other malignancies involving abnormal hematopoiesis or the formation of blood cells. Occupationally, benzene can be found in environments with petroleum and crude oil workers, where they are often exposed in extreme rates. However, benzene is a very common air pollutant, so not only can crude oil employees be exposed to it, but so can the population at large. It is ever present in gasoline, car exhaust, tobacco smoke, industrial emissions, polluted air, and polluted water. And we absorb and are exposed to benzene through inhalation. Scientists can measure benzene levels through urinary phenol bioassays. This allows scientists to conclude that repeated exposure to benzene leads to a higher risk of toxicity and tumorigenesis, and the study was done with crude oil workers. Benzene causes chromosome changes in bone marrow cells, commonly found in human leukemia cells, causing a buildup of lymphocytes in the bone marrow. Through experiments with rats, benzene can also cause tumors of hematopoietic and lymphoid tissues in rats. Next, we will go into understanding the following human mechanisms of how benzene induces acute myeloid leukemia.
When benzene is inhaled into the lungs, it is initially metabolized into benzene oxide, which also exists as oxypin. This can be then converted into hydroquinones, also known as HQ, a chemical species that takes us one step further from the lungs into the bone marrow. Other metabolites that are produced in this process are called 1,2,4-benzene triol and catechol. Altogether, these are very reactive electrophiles that can react with peptides within the bone marrow, interrupting cellular processes. This process is then magnified with increased exposure to benzene. One abnormal process that occurs within benzene-induced leukemia is the creation of oxygen radicals, or reactive oxygen species. They induce DNA strand breaks, point mutations in the DNA, as well as leave damage in the mitochondria. There is also associated dysregulation of the enzyme NQO1. NQO1 is an important enzyme that converts benzoquinone or BQ back to hydroquinone, trying to reverse the toxicity that is occurring within the bone marrow environment. This is all a part of the oxidative stress response of the body. Metabolites are also associated with aneuploidy, or an abnormal number of chromosomes. Specifically, we have observed monosomies in chromosomes 5, 6, 7, and 10, and trisomies in chromosomes 8, 9, 17, 21, and 22. It seems as though the chromosomal loss observed in the bone marrow cells may be due to interrupted microtubule assembly. Another abnormal process seen is the inhibition of the topoisomerase II enzyme, which plays a very important role in DNA replication. When helicase unwinds the DNA, supercoiling occurs at both ends of the still double-stranded DNA, and topoisomerase II then breaks the DNA to unwind it and relieve the stress caused by the supercoiling. Compromised immunosurveillance is another example of dysregulation of the bone marrow. In workers exposed to benzene, we see a noticeable decrease in red blood cells as well as white blood cells. This contributes to converting the normal anti-tumor immune microenvironment to an immune suppressive microenvironment that supports unregulated proliferation. Compromised regulation of an important receptor also characterizes this process. AHR, or transcription factor aryl hydrocarbon receptor, is an important receptor that mediates the hematotoxicity caused in this process. It regulates between the temporary and reversible growth arrest period called quiescence and the proliferation of hematopoietic cells. This dysregulation in the hematopoietic stem cell niche is further characterized by cells with chromosomal loss or damage that can escape apoptosis and detection. This allows for the proliferation of pre-leukemic stem cells and eventually leukemic stem cells. Now, these hematopoietic progenitor cells proliferate as part of tumorigenesis, leading to acute myeloid leukemia.
For chronic lymphocytic leukemia include targeted oral therapies that include a combination of the drugs idelalisib and venetoclax, along with anti-CD20 antibodies like obinutuzumab, which are activated glycosylated phosphoproteins expressed on the surface of B cells. These anti-CD20 antibodies have led to improved outcomes of chronic lymphocytic leukemia in many patients, even those with high risk factors such as mutations in their D17P13s and TP53s or immunoglobulin heavy chain genes like IgHV genes. However, the specific treatment depends on the comorbidities of the patient. For young, fit patients, scientists recommend chemoimmunotherapies with fludarabine, cyclophosphamide, and rituximab because of their curative potential. Chemoimmunotherapy is a combination of chemotherapy drugs and immunotherapy drugs, which stops the cells from growing and dividing uncontrollably. Scientists say the best form of chemoimmunotherapy is monotherapy with alkylating drugs.
From the research that has been conducted by scientists all over the world, some questions are still left unanswered. For example, lymphoid leukemia has been seen to develop in dogs, but the cause is still unknown. From the research we have found, we believe benzene may be the reason. Dogs are man's best friend, which means they share the same environment as we do and come into contact with similar pollutants. We know that due to how benzene is metabolized in the body, there is a possibility of it leading to cancer in dogs as well. There is no direct association of some common AML gene mutations. However, as mentioned before, there is an association between mechanisms that cause DNA mutations and benzene, such as oxygen radicals. With time, we hope more research will be done to gain a better understanding of many unclear mechanisms through which benzene metabolites induce AML and other hemopoietic malignancies.